So we're going to talk about two important vitamins, and this really will set up the last lecture on nucleotides, and I'll want you to really pay attention to them because they're very important, extremely important in metabolism of amino acids, also extremely important in the synthesis of nucleotides, in particular folic acid. Okay? So you want to really pay attention and understand that. So we'll start with folic acid. And I just want to show you that folic acid is what we get from the diet. We cannot, it's a vitamin. That means we cannot make it. So we get folic acid from the diet. We have one enzyme that will convert it to the active form, and the active form of folic acid is called tetrahydrofolate. Again, you don't need to memorize structure or anything, but that's the active, biologically active form called THF. Okay? The enzyme that we have is called dihydrofolate reductase. One enzyme, but we have to get the folic acid from the diet, converting it to this active form. Now I want you to see this highlighted part. Folic tetrahydrofolate, the best way to think of it is that it is a carrier of one carbon molecules. So you have a formal carbon with a carbonyl, the carbon here, double bond there, CH, CH2, methylene, methyl. There are a few other forms. We've already covered this one briefly when we were talking about methionine. There are at least 6 or 7 different forms of one carbons that can be carried by tetrahydrofolate. So in that sense, tetrahydrofolate becomes the donor or acceptor of these one carbons. Okay? That's its function. Very important to appreciate this role of tetrahydrofolate. Note the 3 processes it is involved in. Purine synthesis, purine nucleotides. That means without THF, you cannot synthesize adenine and guanine nucleotides. That ATP, GTP. Without THF, you cannot synthesize thymidine. And without THF, you cannot resynthesize methionine. That's just a few of the THF requirements. But 3 of these are extremely important in terms of nucleotide biosynthesis. Adenine and thymidine are adenine nucleotides and thymidine are extremely important. The reason they're important because without nucleotides you can't make nucleic acids, and without nucleic acids you can't have cell division, replication, and all of that. So appreciate how important THF is. Because of that, THF, because of its importance, THF is one of the most prevalent worldwide deficiencies because of malnutrition in many cases for pregnant women, and it is presumed to have really a role in several birth defects, including this one here, spina bifida, affecting the spinal cord development. Okay, so just again to refresh your memory. So there are 3 things on this here slide: A, B, and C. C is the biologically active form of THF. Okay, tetrahydrofolate. What we need as humans from the diet is this one, B, folic acid or folate. One and the same. Folic acid, folate are one and the same. So you need this. We have one enzyme that converts this folate to tetrahydrofolate. And that enzyme is dihydrofolate reductase. Now, in microorganisms and bacteria, other eukaryotes, they can start with A. They can make their own folate. Okay, that's the difference between us and them. We cannot make this structure. They can. It's a multi-step process. They can start here, make folate, then make THF. So actually, eukaryotes are almost self-sufficient regarding THF. They can make it from scratch. And that's the precursor here, para-amino pteridine. Basically, it really becomes para-amino benzoate, and then it's converted all the way to THF in bacteria only. They can do that themselves. Okay? So here's the important thing. I want you to appreciate first, as I mentioned before, this is where I'm going to start. Both humans and microorganisms need tetrahydrofolate. Why do we need it? Why is it needed? It's needed for purine synthesis, thymidine synthesis, and really several amino acid metabolism. Okay? So very important. In particular, the bottom two here are extremely important because without purine and thymidine synthesis, without nucleotides, you cannot synthesize DNA and RNA, and therefore you can impair growth, cell division. Okay? So this is really an important strategy for some of the therapeutic treatments. In humans, we said we have this one enzyme only that converts folic acid from the diet to THF. Actually, it is the target of an important anti-cancer drug, chemotherapy drug. It has other uses, but that's one of the important chemotherapy drugs called methotrexate. It inhibits dihydrofolate reductase. So there's very little THF produced as a result. If you don't have THF, you can't make purines. If you can't make purines, you simply can't make DNA or RNA. Okay? Purines are ATP and GTP. And this is really an important concept maybe some of you aren't aware of. But in order to synthesize DNA and RNA, you need the 4 nucleotide triphosphates to be available in roughly equal amounts. Okay? For DNA, you need the deoxy derivatives. You need the deoxyribonucleotide triphosphates, all 4 of them. Deoxy ATP, deoxy GTP, deoxy TTP, and deoxy CTP. Okay, so you need all 4 to be present. If one is missing, you cannot make DNA. If you can't make DNA, you can't have cell division. You can't have growth. Okay, so this is the idea that you inhibit this enzyme, you slow down cancerous cells. Cancer cells are constantly dividing. Okay? Now, the same concept but a different target, works as an antimicrobial agent. So as I mentioned, this intermediate here called PABA, para-aminobenzoic acid, can be converted eventually to folic acid in microorganisms, and then microorganisms convert it to THF. Okay? So the strategy for the first actually antimicrobial was this molecule here called sulfonamide, and it inhibits this enzyme dihydropteroate synthase. It inhibits it, prevents bacteria, microorganisms from making folic acid. If they can't make folic acid, they for sure cannot make THF. So you slow down bacterial growth. Okay, so two important targets here. I want you to know what this is the first really antimicrobial discovered back in the 1950s, and very important target enzyme dihydropteroate synthase, and in humans methotrexate, important anti-cancer chemotherapy drug. Okay, so THF is used in a couple of places. It's used in the synthesis of serine from glycine or glycine from serine. Think of it as the acceptor of the hydroxymethyl group. That's it. That's the difference between serine and glycine. Is serine has a hydroxymethyl, glycine has a hydrogen as the R group. So that's all you need to know, is that THF is used there. THF is also used in the degradation of histidine. Here it's the acceptor of the formamino group, this one, and it allows further metabolism of histidine to glutamate. So two amino acids requiring THF, their degradation or their biosynthesis, the biosynthesis serine glycine degradation of histidine. The third one is obviously we talked about before, requiring THF, is methionine synthesis from homocysteine. Okay, we'll get back to it in a minute again. So we're going to go to vitamin B12. Vitamin B12 is very important, and what's unique about vitamin B12 is, in general, or as far as I know, it's only found in animal sources. Although some people will argue that sometimes, but I don't know why. But this is where it's important. So for example, vegans have to make sure they get vitamin B12 from some source if they're strict about their diet. Vitamin B12 is a pretty bulky, large molecule. Okay, you see here chlorine rings with cobalt in the middle. Don't worry about the structure, but it can exist in two different forms. These two are the naturally occurring methyl and deoxy, so don't worry. And this is what's usually supplemented if you're taking it as a supplement. But anyway, vitamin B12 is required for only two reactions in the body. However, they're both extremely important. The first one is the formation of methionine from homocysteine. You remember we covered that last lecture. It's the enzyme called homocysteine N-methyltransferase, also known as methionine synthase. It's remaking methionine from homocysteine. For that reaction, you need both B12 and tetrahydrofolate. If either one of them is missing, you're going to have an accumulation of homocysteine resulting in homocystinemia. Actually, if either one is missing, you're also going to get megaloblastic anemia, which means that the red blood cells don't develop very well. So there's two things that occur simultaneously because of that. The second enzyme here is the isomerization of an important intermediate in the metabolism of several amino acids, methylmalonyl-CoA, which eventually becomes succinyl-CoA. So all the amino acids that give you succinyl-CoA, which are isoleucine, valine, threonine, and methionine. But that intermediate is also derived from odd chain fatty acids. Okay? So actually, before I go to that, let me remind you of what we did last time. Here's homocysteine derived from methionine. It's going to be converted back to methionine by methionine synthase, and it needs in succession methyl tetrahydrofolate first, giving the methyl group to vitamin B12, vitamin also called cobalamin, methylcobalamin. Then vitamin B12 gives that to homocysteine to form methionine. Okay? So they're both needed. If either one is missing, you're going to have accumulation of homocystinemia due to a vitamin deficiency. Okay? So the other enzyme that uses vitamin B12 is this one here. So don't worry about all the details up there. Just pay attention to this one. Methylmalonyl-CoA mutase is the enzyme, and it is going to convert methylmalonyl-CoA to succinyl-CoA. Anything that gives you propionyl-CoA is eventually converted to methylmalonyl-CoA, then succinyl-CoA. As I mentioned, there are the methionine, isoleucine, valine, and threonine all give you propionyl-CoA and eventually become succinyl-CoA. But in addition to that, odd chain fatty acids. So think of a fatty acid that's 15 carbons. If you break it down, the last fatty acyl chain obtained is propionyl-CoA from the beta oxidation of odd chain fatty acids. Propionyl-CoA can in turn be converted to methylmalonyl-CoA, then methylmalonyl-CoA can be converted to succinyl-CoA. Remember, succinyl-CoA is a glucogenic carbon skeleton and can be used to make glucose. So as such, odd chain fatty acids can be a source of carbon skeleton for the synthesis of glucose, unlike even chain fatty acids that cannot. So in summary, B12 is needed by one enzyme required for odd chain fatty acid metabolism, as well as the these four amino acids: methionine, valine, isoleucine, and threonine. Okay.